So here we go with our evolution notes number 4. This is part 2 of phylogeny. Okay, so before we were talking about how organisms are grouped into domains and kingdoms, phylum, class, order, family, genus, species, Linnaeus's classification system. And now we're going to talk about how we could put organisms into clades, which is kind of along the same lines of the taxonomic groups, that they're nested within each other and one leads to the next. So cladistics is one approach to systematics, one way we can study organisms and how they all fit together. And it's going to use common ancestry to classify those organisms. Each organism, or species, excuse me, is placed into a clade, which includes the ancestor and all of its descendants. And we'll talk about the different kinds of clades there can be. Okay, so clades can be either monophyletic, polyphyletic, or paraphyletic. We can have a clade and a taxon being the same thing if it's monophyletic, which basically is a single tribe. So that would be the ancestor and all of its descendants. Okay, it can be paraphyletic, which means beside the tribe, where it has the ancestor and some, but not quite all of its descendants, and we'll look at an example of this in a second. Or it can be polyphyletic, which means many tribes, and it includes taxa with different ancestors. So let's take a look at each one of those. Okay, so in this first one, the monophyletic, we've got the ancestor here and all of its descendants. Okay, so that would be monophyletic. In the paraphyletic, we've got the ancestor here and some, D, E, and F, but not all, G is excluded, of its descendants. And then finally, we've got the polyphyletic group where we have an ancestor here, but we've also included organism or species C over here, which has this ancestor or that one. So it includes this ancestor and all of its descendants plus another ancestor. Okay. So when we talk about characteristics and ancestors, we can look at what are ancestral characters and which ones are derived characters. Okay. So we know that organisms share some characteristics with their ancestors, but we also know that they have differing characteristics from their ancestors. The shared ancestral characters are those that were in the ancestor, or originated in the ancestor, but may not include all of the organisms. Okay? So for instance, backbones in mammals. All mammals have backbones, but not just mammals have backbones. So that goes all the way back to that common ancestor, whereas a derived character is unique to that particular clade or group, which would be hair in mammals. Okay, so hair is something that all mammals have, but not all animals. Okay, and this is relative to what you're actually looking at. So depending on the characteristic that you're looking at, depending on the clade that you're looking at, it could be ancestral or derived. It just depends on what it is that we're trying to figure out. So those derived characters are unique, and we know that they arose from the ancestor, even though the ancestor may not have that. Okay. By examining these shared derived characters, we should be able to determine when each one appeared and the evolutionary relationship to the whole group because of it. So let's take a look at how we would put these together, how we put together these cladograms, or things that we've looked at before. And we have to decide first which are going to be the in and the out groups. The in groups are the group which has the species we're studying. Okay, so in, our example here, make this bigger. we have leopards, turtles, salamanders, tuna, lampreys, and lancelots. Okay, the outgroups are a group that diverged from the lineage before the group we are studying. Sorry for all the movement there. And that would be the lancelots, because they are the outgroup. They don't have an actual backbone. They're chordates. So let's take a little bit. Okay, so let's take a little bit of a closer look. When we have an outgroup, that means that that group doesn't have any of the characteristics that we're studying. So the lancelets don't have hair, they don't have eggs, they don't have legs or eggs, they don't have a hinged jaw, and they don't have a backbone. Okay, and then we're going to take a look at some other organisms, and basically, hopefully what we'll end up doing is adding one or two characteristics at a time. So you can see by the color-coded, as we go up and we look at these different organisms, we see that they're getting more and more complex each time. So our in-group was the leopard. That's what we're studying. And by creating one of these charts, we can actually see how the characteristics evolved. Okay, so for branch lengths, sometimes it doesn't matter how long they are, and we have to remember that the chronology is relative. It's more of an earlier versus later instead of exactly how many millions of years is occurring. But some diagrams actually have branches that will reflect the number of changes over time, and they can draw on fossil evidence to come up with those. So let's take a look at what that looks like. Okay, so here we have a diagram that actually shows the number of evolutionary changes, and it's over time. So we have our ancestor back here, and then this is showing time. So we've got like 1 million years here before we get another characteristic, 1 million here. This little section here is 500,000. Okay, so it's just showing you how long it was in between each group of organisms until the next characteristic evolved. But that doesn't always happen. Okay, in some trees, the length of the branch is going to indicate the number of changes. So the longer the branches, the more changes to the next organism. Okay, so by looking at this, we can see that there are not very many changes between humans and mice. Okay, but if we were going from a fruit fly, which would definitely be our outgroup, to the zebrafish, we've got lots of different changes to get to that point. One thing that we can talk about when we're trying to build these is parsone. The more DNA that we study, that's awesome, but the bigger our database grows, and the more difficult it is to build these trees, these branching diagrams. If we are just looking at 50 species, we're looking at 3 times, this should be a superscript, 10 to the 76th ways to arrange those organisms in a tree. So we can never know for sure exactly what it is, 100%, but we can narrow the possibilities by applying something called maximum parsone. Basically what that's saying is investigate the simplest explanation first, the one that would require the fewest evolutionary events or fewest base changes in the DNA, and then we can go from there. Okay, so let's kind of take a look at what one of these looks like. Remember that trees are hypotheses, but let's look at 3 different hypotheses that we can have. We can say that either species 1 and 2 branched off from each other, and species 3 was on its own, or species 1 and 3 are most related and 2 was on its own, or species 1 was on its own and 2 and 3 are more closely related. So just because we can already see it over here, let's take a look at this first hypothesis. It says that there were 6 events that happened. Well, let's take a look at the ancestral sequence and how many events had to happen between that and 1 and 2. Well, we know that there are 3 because A, C, T, G, T, A, and T, T. There's 3 different events that had to happen. Well, if we say that also for species 2, there were also 3 events that had to happen, we can put those together and say that 2 of those events happened to both of them, and then 1 event is different, and that's where they split off. Whereas species 3 just has 2 events that changed it from its ancestor. Okay, so these 2 are the same, these 2 are not. So that's how they came up with that hypothesis. Okay. Like I said, a tree is a hypothesis about relatedness of organisms. We try to get the best hypothesis, which is the one that is best fit to all the data that we have, but, and it frequently happens, they are modified as new evidence comes forth. We can use those to try to make and test predictions. Now we're going to talk about molecular clocks. Okay, so some genes or regions of the genome seem to be evolving at a constant or a consistent rate. What we're assuming when we look at this idea of a molecular clock is the number of nucleotide substitutions, T to A, T changes to G, whatever, is related or is proportional to the time lapsed. So we have the common ancestor, and there's 6 changes that should be a certain amount of time. Okay. What we can do is then graph the number of genetic differences against the dates of things that we know from the fossil record, and then hopefully use those dates to estimate other dates that we couldn't get from the fossil record. So here's an example of one of those nucleotide substitution graphs. We have the number of nucleotide substitutions against millions of years. So you can see the fewer the substitutions, the less amount of time it took. Whereas all the way up here, the kangaroo, 120 some million years, and over 100 or close to 100 nucleotide substitutions. There are genes that evolve more slowly or more quickly than others, and some genes evolve millions of times faster than other ones. So we have to remember, like with everything else we've talked about, it's all relative to what we're actually looking at. Okay? Some mutations can be selectively neutral, but many are harmful and they're eliminated quickly. So that's going to determine the rate of change. The more critical the gene is to the organism being able to survive, the more slowly that organism will evolve, because changes to that gene can cause death. So the least critical are the genes that evolve the quickest. So there are some problems with using the molecular clock and molecular clock assumptions, and that is that they don't run as smoothly as expected because we have natural selection, which favors some genes over others because of the particular environment at that particular time. They're not really certain after about 550 million years ago, so we can only use it for, relatively speaking, recent times. And then if we try to use multiple genes, we can actually improve the accuracy of our molecular clock. Just one doesn't really tell us much, but if we use a whole bunch of them, we can learn a little bit more. So now we're going to see an example applied to humans of how this can work. Okay, so we've got human immunodeficiency virus, And we can actually use a clock, or we used a molecular clock to date the origin of HIV infection in humans to figure out when was it that humans first became infected with HIV. So to do this, there's many different strains of HIV, but they looked at HIV-1M, which was the most widespread strain. They were able to compare samples from many different years that they had and use that, plot it on a molecular clock to figure out that it did show a clock-like evolution, that every so many years there was so many sequence changes to the HIVs. So by this, they were able to calculate that HIV-1M first spread to humans somewhere in the 1930s. So not exact, but pretty close. So what we can do is put this all together and see how things may have evolved. So this is one example of how organisms could have come about from that common ancestor. And you see we have our 3 domains. Okay. Right now we have, if you'll notice in here, we don't have anything that's specifically called the protists or protozoans. It's all now divided out into different groups of organisms or different clades. So protista is not really recognized as a kingdom anymore. Okay. And then we have the bacteria and the archaea. It looks nice and neat and pretty and looks like everything fits together. But then if we really take a look at what is going on and in our using our molecular clocks, we know that there's a lot of horizontal gene transfer that's happening, especially between lower organisms, okay, the prokaryotes, the ones without a nucleus. We know that genes are transferred from one species genome to another through pieces of DNA called plasmids, through viral infections, fusions of 2 organisms coming together. And when we study those prokaryotes, we can see that about 80% of the genes have moved between species. So that kind of puts us in this like crazy, not even a tree anymore, but this like tangled web of roots where we have ancestral populations giving rise to all kinds of differing branches that are maybe converging or diverging. Okay, so the question is, was it the one that we just looked at, or is it more like this, and this is how life all came about?